And I got the honor of being the first speaker of today, and to kind of briefly introduce some of the things that we're going to be discussing this afternoon. The title of my talk is The Comparative Pathogenesis of Psoriasis and Atopic Dermatitis. It's hard to give this justice in just 15 minutes or so, but I'll do my best. These are my conflicts of interest. And then, I find it to kind of be very helpful to kind of just put up a photograph of psoriasis and atopic dermatitis. And in some ways, you can see how different these diseases are, but at the same time, you can also see how similar they are. They can be pretty extensive. The skin is red and inflamed, but you can also see, these subtle kind of differences between the diseases, the thick kind of silvery scale of psoriasis, and then the more kind of wet-like appearance of atopic dermatitis with sometimes marks of excoriations given the intense pruritus that is associated with this disease. I know the group here is very familiar with psoriasis, but it's always good and helpful to kind of revisit some of the features of psoriasis. So there on the right, you have some clinical photographs that I have taken over the years of cases that I've seen in the clinic, and just gives you an idea how variable this disease actually can be. It's a common disease. It affects about 2% of the US and European population. It's a disease that is genetically predisposed but complex, and that means that there are multiple different variants that contribute to disease susceptibility. And to date, we have identified about 160 of those. The age of onset of psoriasis is usually in adolescence or young adult and tends to persist throughout life. The disease pathogenesis incorporates both mixed autoimmune and innate immune responses. And in about 20 to 25% of patients go on to develop psoriatic arthritis, which can be quite debilitating. And then there are multiple other comorbidities associated with psoriasis, including cardiovascular disease, metabolic syndrome, including type 2 diabetes. What about atopic dermatitis? It's one of the most common inflammatory disease of the skin. There's about 15 to 20% lifetime prevalence. It's also a complex genetic disease, multiple genetic variants, each one with a fairly small risk, but combined, they really help to kind of drive the disease pathogenesis, and we are up to over 100 genetic loci known to predispose to atopic dermatitis. And if you look at what those genes or the genetic variants are doing, they're really contributing and incorporating defects in the epidermal barrier, activation of different T cell subsets, and then there's dysbiosis of the commensal skin microbiome associated with this disease. It's a very heterogeneous disorder, variable clinical manifestations, and includes both acute and chronic presentations. And in contrast to psoriasis, the comorbidities that are associated with atopic dermatitis tend to differ quite a bit, and that includes food allergies, asthma, and allergic rhinitis being the most common ones. So if you think about, the histology of these two diseases, these are both papular squamous disorders, and that means that they're both involving the epidermis and abnormal epidermal function. And you can see that the changes on the plaque psoriasis end tend to be a little bit more and better defined than you can see in atopic dermatitis. But the immune networks that I have there on the bottom tend to be a little bit different, with psoriasis being kind of centered on IL-17, IL-23, and TNF-associated inflammatory mediators, and atopic dermatitis being different with IL-13, IL-22, and IL-31 being kind of the key ones. My background is in genetics, so I often kind of revert to genetics as a way to kind of look at diseases, and this is a kind of a diagram that kind of links together the different genetic variants identified with psoriasis with other chronic inflammatory diseases. You have psoriasis there in the middle, and then the different risk genes are kind of shown with smaller letters. But what I wanted to highlight is both the overlap of psoriasis with multiple other immune and autoimmune diseases, but it also has a bit of an overlap with atopic dermatitis, particularly involving the IL-13, IL-4 locus. And Dr. Brown is going to be telling you more about this in her talk later this afternoon. But I find it to kind of be quite interesting. If you look, this is what they call like a Manhattan plot of psoriasis. And you have the psoriasis metageos over here, and then you have the atopic dermatitis metageos over here on this end. And this is an old study. It was published about 9 years ago. But you can really see the peaks that you have in psoriasis sometimes kind of modeled a little bit on the atopic dermatitis end. Maybe not as strong. Here's the epidermal differentiation complex, much stronger in AD compared to psoriasis. and here you have the IL-13 genetic locus. But if you combine them, you get a very significant peak right here involving the chromosome location 5q31.1. This is the IL-13 gene locus. And it's actually interesting to think about it that in atopic dermatitis, you have an odds ratio with this locus of close to 1.3, but in psoriasis, you have the opposite. It's actually protective, about 0.86. So this is the conclusion that these authors made, that the results show that atopic dermatitis and psoriasis have distinct genetic mechanisms with opposing effects and shared pathways influencing epidermal differentiation and immune responses. So the variants predisposing to psoriasis tend to be protective for atopic dermatitis and vice versa. So it's this kind of like a mutual antagonism between these two diseases. This is a study that my group published about 7 years ago in the Journal of Investigative Dermatology, and it's based on bulk RNA-seq data. So what it shows you is that we're just comparing the two diseases on the transcriptomic level. And if you look at psoriasis versus healthy and then atopic dermatitis versus healthy, whether it's the upregulated or the downregulated genes, you can appreciate that there's much greater number of differentially expressed genes in psoriasis compared to atopic dermatitis. Perhaps not surprising if you look at the degree of epidermal changes that happen in these two diseases. And then you can kind of compare the overlap between psoriasis and normal skin, normal versus atopic skin, or psoriasis lesional versus atopic lesional, and you can appreciate the overlap, and then how the genes in psoriasis correlate with the ones that are differentially expressed in atopic dermatitis. And you can appreciate that it's actually quite similar. There's almost like a linear kind of a graph going over here. It's a little bit more skewed towards the psoriasis side than the atopic dermatitis side, but overall pretty similar. And if you look at the differentially expressed genes, about 81% of the genes that are differentially expressed in atopic dermatitis overlap with those of psoriasis. If you look at it from the other end, only about 41% of the genes that are differentially expressed in psoriasis overlap with those of atopic dermatitis. So these diseases have a lot in common if you look at it from the transcriptomic angle. What about the cytokine responses? There are definitely antagonistic immune profiles in psoriasis. This is a study of about 147 individuals. And you have the psoriasis lesional here in green, and then you have the atopic dermatitis here in the purple, and both lesional and non-lesional skin, and then healthy controls right here in the other corner. But what you can see is that the IL-17 response is what really defines and separates psoriasis from atopic dermatitis, where IL-13 responses dominate. And this kind of mutual antagonism actually is well described. This is a paper that was published in New England Journal of Medicine about 13 years ago from the IRIC group, and it's a patient that actually has both psoriasis and atopic dermatitis. This is the histology between the two. Here you have the psoriasis lesions, and then more like atopic eczema, atopic dermatitis. And then you can appreciate the clinical course where you're looking at atopic dermatitis and then psoriasis here in blue. And you can see that when one disease is flaring, the other one actually is getting better. And this is kind of like part of the spectrum that we often see with these patients, extremely rare, but we see them from time to time. And then you can see the histology is also quite different. IL-17 is much higher in psoriasis, and there's a little bit of it in atopic dermatitis. And you can see the same thing with IL-4 and IL-22, with IL-4 being more in atopic eczema and interferon gamma being more higher in psoriasis, and IL-22 kind of being high in both, maybe a little bit higher in psoriasis. But it's also kind of important, and I want to highlight that, and we will have a talk on this also, is that the immune network in both atopic dermatitis and psoriasis actually is quite plastic. This is not a static kind of a diseases. There is quite a bit of shift, and we can see this when we treat patients with these very targeted kind of a biological therapies. And it's well known that if you use either anti-TNF, anti-IL-17, or even anti-IL-23 agent, that a proportion of patients may actually go on to develop eczema-like lesions. And we're seeing more and more of this also on the other end. We have patients with atopic eczema or atopic dermatitis being treated with dupilumab, or even anti-IL-13 treatment, is that you begin, at least in some patients, to see shift towards more like a psoriasis-like phenotype. So it is quite dynamic, and this is still not well understood, but the mechanisms involved. We have looked at this in our lab, this is based on a single cell data. You can look at the different immunological network. And the tools that we have today to kind of study this have really exponentially increased our chances to kind of take a really deep dive into the pathology. So if you look at CCL17 and CCL22, here we're comparing atopic dermatitis in red and psoriasis in green. You can see that these two cytokines or chemokines are higher in atopic dermatitis. But the ones that drive Th17 responses, such as CCL20, which is a chemokine for Th17 cells, or CCR6, which is the receptor on those cells for CCL20, that's much more dominant in psoriasis. You can also do the same thing for Th2 signal. You can look at IL-13, very strong in atopic dermatitis, barely detectable in psoriasis. But interestingly, the receptor for IL-13 and IL-4 are much more highly expressed in psoriasis skin. And what does that mean? We just don't know. And then also, if you look at the cell-cell interactions, this is just limiting it to the dendritic cell T-cell interactions. You can see the Th2 signal or ILC2 signal here is much stronger in atopic dermatitis and barely active in psoriasis, where the shift actually is towards other immune cell subsets. You can also look at this. This is just focusing on the dendritic cells. We have 12 subclusters of dendritic cells. You can separate that into 5 major clusters of dendritic cells, and then you can overlay the disease states to kind of see how they're kind of defined. You can see there's a little bit more psoriasis on this end and a little bit more atopic dermatitis on this end. These are the marker genes that you use to define these subsets, and then you can look at the cell numbers of the individual dendritic cells and see where it's actually skewed. And you can see that for the conventional dendritic cell 2A, they're much higher in atopic lesional skin compared to psoriasis. It's certainly a lot higher than you can see in non-lesional or control skin. You can look at them, and then you can use these data to kind of dig in even deeper. You can look at the differentially expressed genes in a particular cell type, which ones are increased or skewed towards psoriasis, and which ones are skewed towards atopic dermatitis. You can look at their expression of inflammatory mediators, as shown here in psoriatic skin, and particularly with activation of NF-kappa B and IL-8 or CXCL8. And then in atopic dermatitis, you're seeing much more of the CCL17, CCL22, and CD1B expression. And then you can use the tools that we now have to really kind of see how these dendritic cells are actually behaving differently and derived differently. So you have, these are all the dendritic cells in both psoriasis and atopic dermatitis. Here we have a conventional dendritic cell 2A, and here 2B, and then you have an intermediate state. And we can see that it's actually quite a bit of a trajectory where you have the CD2As on this end and the DC2Bs on this end, and then you can kind of compare how the different chemokines are actually separating on this. You have atopic dermatitis here, psoriasis, lesion control. So a lot more dendritic cells in atopic dermatitis. The chemokines are really expressed here on the atopic dermatitis, the DC2A end, whereas the IL-8 and IL-1B is much more on the psoriasis end. And then you can see how these dendritic cells are really diverging in terms of their immune activation in these 2 different disease states, where it's going up in atopic dermatitis and down in psoriasis. And then you can look at differentially expressed interactions, looking at the ones that are enriched in psoriasis versus the ones that are down in atopic dermatitis. And you can really see that it's the DC, dendritic cell interactions that are really shifting towards being more active in psoriasis and shifting the balance with atopic dermatitis having more of a DC2A function and psoriasis more heavy on the DC2B end. And that's kind of shown over here. You can look at the different interactions. I'm not going to go into the details, but you can really see that the interaction between the two is really shifted between psoriasis and atopic dermatitis. And we think that this is one of the reasons why you're seeing this different polarization patterns in these 2 diseases, and also where you can have the switch between the 2 in these paradoxical type reactions. So just to briefly summarize, the psoriasis and atopic dermatitis are 2 of the most common chronic inflammatory skin diseases. Both have overlapping histologic features and transcriptome, but they're characterized by opposite polar cytokine patterns. And while they're typically mutually exclusive, both immunologically and genetically, there are rare patients that have features of both atopic dermatitis and psoriasis. And the response in these diseases actually is quite plastic. And we can see these shifts with targeted therapies, but the mechanisms of those shifts are largely unknown. And then the regulatory mechanisms involved in regulating the polar T cell responses are largely unexplored, but dendritic cells based on some of the data that we have, may play a key role in that part. Last but not least, I want to thank the IPC and the ISC for being here today and showing you some of the data. And then last but not least, I also want to thank the different funding sources that I've had over the years that have supported my work, and then my lab members and colleagues that I've worked with and collaborated on this. So thank you all for listening, and I think we may have some time for questions.
That's great. Thank you very much, Johann. We've got time for questions. Could I please ask that those who wish to ask a question identify themselves and their institution and country would be helpful. I can't believe there's no questions, so I will kick off. And it's fascinating, this single cell analysis. It looked a little bit heterogeneous in some senses, and I wondered whether you've been able to think about the different kind of subsets of psoriasis and AD that we think about, whether filaggrin-driven disease looks different to not, and so whether that alters the dendritic profile in that context.
Yeah, no, I agree. That's the problem with them, is that the level of heterogeneity, and that certainly is a lot bigger in atopic dermatitis than psoriasis. We see psoriasis being kind of less diverse. That being said, if you go into the clinical subtypes of psoriasis, you begin to kind of see these drifts, and some of them actually extend on a spectrum between an adaptive and more on the autoimmune innate type inflammatory responses. I think nobody has really compared, at least none that I know of, compared the filaggrin positive versus negative individuals in terms of like single cell type analyses. And I think that needs to be done. And also what we really need for practically both diseases is to try to link the genetics with the biology. There's still a major gap between our understanding of the genetic predisposition of those diseases and how those genetic variants actually play a role in the disease mechanisms. We don't know what cell type actually is primarily involved. We don't know if the genetic variant is activating those cells or making them more active or less active. And how the interplay between that. But we now actually have the tools to really dig into that. And I think that's where the field needs to go. But those types of studies are very expensive to run. And you need, 200 or 300 patients to do it well. So I think it's coming, but we're a little bit off. And I think that's going to address the heterogeneity, because it's not just molecular heterogeneity, it's also genetic heterogeneity.
Yeah, and phenotypic. So I think one of the issues with this data, which we should encourage the community, is to be able to properly give the clinical phenotypes associated with the molecular data. Yeah. Because that's been limiting. I think everyone puts their data in relevant repositories, but there isn't always access to clinical phenotype.
Yeah, I agree. Access is critical.
Okay. Any questions then from the floor? No? If not, we'll thank Johann again. Thanks a lot.